but we're rolling with it, and all is good now. We have Anna here. I want to introduce the webinar. I think Kyle is our guest host today. And Kyle, do you need video, or are you good to go? You don't mind just doing it without video?
Not at the moment because I'm in the car. Unless people want to see me, want to see me driving.
Eyes on the road. Eyes on the road.
I was very committed to MC. So our guest MC, I'm going to turn over to Kyle right now to sort of introduce Anna, and then we'll get rolling with Anna's presentation.
Sure. So it's my pleasure to introduce Anna Gloyn, who's the speaker for the webinar today. Anna's a professor of pediatrics and genetics at Stanford, formerly at Oxford. Anna's research concerns genetics of diabetes, and has done a lot of great work, and in particular elucidating mechanisms of how risk variants for diabetes function. Yeah, so I guess I'll stop there, and welcome, Anna, and thanks for joining.
Excellent. Thanks, Kyle. A couple things. So I'll be monitoring the chat. And before you dive in, we're trying to figure out people think the chat is disabled. So put in the Q&A, and as thank you for noting that. I will try and fix the chat offline with Mackenzie. But please put things in Q&A, and we will try and get the chat handled as we go. But I don't want to take any more time away from Anna than we already have. So
yeah. Now, do we usually stop to answer questions, or are they typically saved till the end?
Typically, I find it's nice, if it's okay with you, Anna, sometimes if they're relevant to where we are, like on a given slide or content, we kind of stop and ask to, like, sort of make it interactive. Is that okay with you, Anna?
Absolutely, although I'll find it very difficult to stop in the flow. So Noelle, you'll have to shout and tell me, Anna, we have a question.
All right, we'll do that.
Because it's hard when you can't see faces.
All right, super. All right, so I will fix the chat, or otherwise use the Q&A for now, and let's get going.
Okay, well, good morning, everyone from North California. Thank you so much, Kyle, Noelle, and the Portal team for the opportunity to deliver one of these seminars. I'm delighted to have the opportunity to showcase some of the work that we've been doing over many years, starting a little bit of this is from my time in Oxford, and then work that has continued since I moved to Stanford. So as Kyle alluded to in the introduction, I've been working in this space for quite some time, and the consistent focus of my work has really been to use DNA variation that we know is either causally implicated in causing monogenic forms of diabetes, or more recently, common genetic variation that increases your risk of developing type 2. And the premise really is that we use these DNA changes that we know are causally related to diabetes as tools and instruments to delve into the underlying mechanisms that cause diabetes. And my lab is particularly focused on the pancreatic islet. Occasionally we spill into other diabetes-related cells and tissues, but our primary interest is around this wonderful microorganism, organ that is responsible for producing insulin. And like most of us who are interested in the genomics of diabetes, we really hope that in addition to increasing our understanding of the underlying pathophysiology, that there will be some clinical translational relevance of our findings. And this might be through the identification of safe and effective targets for novel therapeutic development, or maybe an ability to stratify patients so that we can deliver the most appropriate medication to them that's most effective for their variety of diabetes. So I'm very fortunate because early on in my career as a postdoc working with Andrew Hattersley in Exeter, I was part of a team that identified the most common genetic etiology of a rare form of diabetes that presents in the first 3 to 6 months of life. And this is called neonatal diabetes. And I was able to establish that heterozygous activating mutations in a key component of the machinery that couples glucose metabolism to insulin secretion were a major cause of neonatal diabetes. And on the back of work from giants in the field like Dane, Professor Fran Ashcroft, we were able to understand very early on what the molecular mechanism was, which was an inability to respond to ATP generated from metabolism. And because we already understood that this channel was the target for a class of drugs called sulfonylureas, we were very rapidly, with the work from Ewan Pearson, able to try patients out on sulfonylureas and transition them from an insulin injection to an oral medication with an improvement in glycemic control. So this is a really nice example of how human genetics can take us to safe and effective targets for therapeutic development. After all, we already knew that this was a drug target, and how we can use human genetics to personalize medicine and to be able to deliver the most appropriate treatment to a patient. It's very timely because Andrew Hattersley and Maggie Shepherd have recently put together a podcast that is really sort of reminiscing on this journey of gene discovery and talking about the impact that this has had on patients. And if you're interested, I would encourage you to scan this QR code and visit the podcast website, and you can listen to a first episode from one of the parents of one of the first patients that we identified with this etiology. And then I was fortunate to be interviewed in the second podcast, and Ewan has just released his podcast this week, which talks about the transition to sulfonylurea therapy. So well worth a visit if you're interested in learning a little bit more and understanding different perspectives on this story. So accompanying you today on the journey that I'm going to take you through on our work on digging for gold in human genetic data, you're going to see some beautiful illustrations of female scientists. We started off at the beginning on my opening slide with some wonderful artwork from Rachel Igontowski of wonderful, impactful scientists from history, all female, who've contributed to our understanding of many different realms of science. But we, of course, like to look forward and to think about the next generation. And throughout this talk, you'll see illustrations of what pupils from Headington Prep School in Oxford in the United Kingdom think a scientist should look like. There are some wonderful illustrations from these young girls around the ages of 6 to 8, and I hope you enjoy their artwork as we go through. So I'm going to start by just taking a moment to congratulate the field and to look at the journey that we've been on in terms of how much we actually understand now about the underlying genetic landscape for type 2 diabetes. This is a figure that we put together for a recent review article that's just now been accepted. And I think it's worth pointing out to the trainees who may be listening in that this is a classic example of your figure being out of date before the paper comes out, because there's wonderful work coming out of a collaboration between groups around the world that has now identified a further 145 signals. So taking a moment just to walk through this, you can see that over time we've managed to increase the number of significant loci and independent signals. And for context, you'll see over here on the left-hand side of my slide, there's a little me here back doing my PhD. And that's because one of the things that I worked on during my PhD was a candidate gene study and the identification of variants in KCNJ11. So it shows you that we've gone from candidate gene studies working on one gene at a time all the way through now to working genome-wide, largely in European populations, but increasingly seeing representation from non-European populations. So we've got around 530 significant loci and just shy of 670 independent signals. So a wonderful toolbox to play with. And a shout out to the portal's Jason Flannett for help with this figure, because Jason had made an earlier iteration of this, which made it possible for us to update it recently. So thanks to Jason for that. So given that enormous wealth of information, why is it so difficult to go from those genetic discoveries to improving our understanding of the underlying biology? And really the explanation for that is that we have a huge challenge to overcome, because unlike the protein coding variants that I identified in children like Lily that cause the monogenic forms of diabetes, the vast majority of the signals that have been identified for type 2 diabetes sit in the non-coding regions of our genome and are presumed to have an effect on gene regulation. And that means that when we have a SNP, we have the complexity of trying to understand whether or not it is altering regulation of gene A, gene B, or gene C. Or it could, for example, actually affect more than one of these genes. And it gets even more complicated because the regulation of gene expression is highly context specific. So it matters where in the body this is happening, when is this happening? Is it during development or adulthood? And does it have to occur under a particular stimulation? So all of these questions on which gene the SNP is regulating have to be thought of through the lens of which tissue, when, and under which stimulation. And this means that getting to this underlying protein is quite a tall order. So what I thought I would do today is really showcase 3 different approaches that we've taken in our lab to try and drill down on causal variants for type 2 diabetes, and then how we use those variants to get to underlying pathophysiology for islet cell dysfunction. So I thought I'd start off by telling you about one of the shortcuts that we can take, and that's where we use coding variants as molecular signposts. So coding variants that are causally implicated in diabetes risk that take us immediately to the protein that we want to study. And today I'm going to share with you some recently published work on coding variants in the PAX4 gene that are associated with diabetes and islet cell dysfunction. I'm then going to switch gears and move from working on an individual locus to telling you about some of the work that we've been doing to kind of ramp up and accelerate our ability to understand what happens when you perturb genes in human beta cells. And I'll be walking you through the data that we've published this year on cellular phenotyping at scale in human beta cells and how we've integrated that with genetic data. And then I'm going to wrap up my talk at the end with some exciting emerging data from our lab, where we've been capitalizing on a unique islet resource that we have established with collaborators at the University of Alberta in Edmonton. And I'm going to present data on a GWAS in a dish, where we've been identifying novel regulators of insulin secretion. And my goal here is by showing you these 3 different varieties of approaches that we take, that you'll get an understanding of the different ways that we can work with the community and colleagues to really leverage the genetics to improve our understanding of islet cell function. Okay, so let's start with part 1, which is using coding variants as molecular signposts. And I'm doing this through the lens of insights into islet cell function from genetic variants at the PAX4 locus. So we've been interested for a long time in taking this sneaky shortcut of using causal coding variants to take us directly to the protein so that we could accelerate our efforts in the lab. And we were fortunate to be part of a collaboration that was published back in 2016, a collaboration that many involved in the Knowledge Portal were also critical team members of. And this study was one of the first to look at scale at getting a complete picture of coding variation by performing exome sequencing in 13,000 individuals, about half of whom had type 2 diabetes. And importantly, and particularly for this study, they were drawn from different ancestries. And up here on this Manhattan plot, you can see the genes in which coding variants have been identified. And what I wanted to firstly point out was the fact that this has been an area that we've put considerable effort into. If you like, we've made it our business to study the function of causal coding variants. And you can see the ones here shown in green, yellow, and grey are the ones that we've worked on in our lab. And what you'll note is that there are different types of protein here. There are transcription factors, there are ion channels, there are zinc transporters, there are enzymes, and there are regulatory proteins. So a vast variety of different proteins that are playing a role in diabetes risk. But today, the focus is going to be on this transcription factor, PAX4. And this is a really fascinating signal because it's specific to one population. This was exclusively found in the East Asian population. It's a coding variant where an arginine residue is changed to a histine. And you can see here from the odds ratio that it has a pretty impressive odds ratio for a type 2 diabetes signal. Now, PAX4 is a transcription factor that we know from elegant studies in murine models is required for beta cell differentiation. And importantly, if you lose both copies of this transcription factor in mice, the mice die by day 3. So in mice, beautiful work that's come out of a variety of groups over the years has shown that PAX4 is a transcription factor that is critical for determining the cell fate. So in mice, the endocrine progenitor cells make the decision to become either an insulin-producing beta cell or a glucagon-producing alpha cell through the relative contribution of 2 transcription factors, and PAX4 being one of them. And PAX4, if you like, is the signal to become a beta cell, and ARX is the signal to become an alpha cell. But we know from the work of many that there are substantial differences in the behaviors of rodent islets and human islets. This is at the level of the architecture, which is very different between the 2 species. We know that there are different compositions of ion channels and glucose transporters between mice and humans. We know that there are differences in the regulation of cell cycle control. And really critically, we know that the performance and behavior of transcription factors in islet cell development is different between mice and men. And this has been seen out in many human genetic studies, and notably around the GATA6 transcription factor. So we can't automatically extrapolate this finding from mice to humans, and we need to know if it is true. Is PAX4 important for determining cell fate in humans? Is it the switch between an alpha and a beta cell? So I want to introduce you to a wonderful collaborative team that have spent many years working on this locus and have taken a variety of different approaches to assess what's going on and how this variant is influencing diabetes risk. Now, the variant is specific to the East Asian population, so we were incredibly lucky to be able to collaborate with Tai Shong and Adrian Teo from Singapore. And this work was led by 2 wonderful early career scientists, Nicole Krentz, who I'm delighted to say is now an assistant professor at UBC and has set up the Krentz Lab. And Huey Lau, who's now Dr. Lau. She, at the beginning of this, was a PhD student. And I think this takes you through the amount of time that went into this study, because we have 1 postdoc to faculty transition and 1 PhD to postdoc transition. So what did the team do? Well, we took this variant that had been identified in the PAX4 gene, and we decided that we would comprehensively assess what it does to a pancreatic beta cell. And we would do this by studying people with the variant and evaluating its effect on their human physiology. We would work with an isogenic cell line, an isogenic stem cell line that we would be able to use CRISPR to knock out the PAX4 gene in. We would collect fibroblasts and reprogram them to iPSCs from people with the mutation from Singapore. And we would use a human fetal-derived beta cell model, the EndoC beta H1, which is more mature, to study the impact of PAX4 later in beta cell development. So with a team from all of our groups and led by the individuals on this slide, we approached what PAX4 R192H does and how it influences beta cell function and diabetes risk using this comprehensive study approach. So first up, let's look at what happens when we study individuals with this R192H variant. So this was performed with frequently sampled glucose tolerance test in Singapore on just over 180 individuals. And as you can see from the data derived from the glucose tolerance tests, there is evidence that if you have the R192H variant, you have elevated fasting glucose, elevated 2-hour glucose values, and a decreased area under the curve for insulin. So really nicely demonstrating that the individuals with this variant have reduced beta cell function. So that really made sense then to think about what was happening in the pancreatic beta cell. What was this change in PAX4 doing to our ability to make a functional beta cell? So we started off by taking an isogenic stem cell line, a line that we use a lot in our lab and have a great deal of data on. And we thought we would use CRISPR to remove the PAX4 transcription factor so that we could study the consequence of loss of this transcription factor on your ability to make a pancreatic beta cell in a dish. And as you can see here from the graphs, we were able to show that we were able to knock out successfully the transcription factor. You can see reduced expression in blue at definitive endoderm, pancreatic endoderm, and endocrine progenitor cells, and also at the stem cell-derived islet stage. And then we were able to take RNA from these cells and to perform RNA sequencing to see what the effect was on the transcriptional profile of the cells in the absence of this critical transcription factor in mice for beta cell development. And you can see over here on the right-hand side of my slide that we were able to show that there was a de-repression of alpha cell genes such as FEV1 and glucagon in the absence of the transcription factor PAX4. So that was in an isogenic line, and we were interested to see what would happen if we actually took cell lines from people with the variant. So took fibroblasts, reprogrammed them to iPSCs, and then differentiated them in a dish. And here shows the results of a variety of lines from individuals with the 3 different genotypes, where we were able to demonstrate that when we went all the way through that differentiation protocol to the end to make the stem cell-like islet cells, that individuals who had the diabetes risk variant had reduced amounts of insulin in the cells that we made, and they had more bihormonal cells. So they had cells that were positive for both insulin and glucagon. So it shows here that the type 2 diabetes, hi, Noel?
Yeah, we have a couple just comments and questions from Klaus, and I don't know if we can allow him to talk, but I will I'll handle it. I think we did this as a webinar. So Klaus mentioned that it's good to see that the mouse model has shown that the mechanism of how PAX4 impacts islet cell dysfunction, differentiation, excuse me. But he did ask, why is there some PAX4 MTA left in the mutant SC line?
Why is there some, in which slide? Which slide are we talking about? This slide?
I tried to do this fast. I think it was, well, it was right, like it was just happened a minute ago. So Klaus, can we unmute, can you unmute yourself? Maybe I can unmute him. I think I can. Hold on, I can do it. Klaus, if you want to talk, I can unmute you right now. There you go.
Wow, thank you. Very kind. No, it's a previous slide during the differentiation time course, where you go show EP lines. There you go. PE and EP seem to have some transcript of PAX4. How can that be? That's puzzling.
Okay, so that's because when you do the CRISPR, you end up making a frame shift, and then you end up with nonsense-mediated decay to remove it. So we don't see any PAX4 protein, but this is at the transcript level, and there'll be some transcript that is picked up at a low level, but it doesn't make it to protein.
Okay, thank you very much.
You're welcome. Okay, so I got to the point of telling you that we'd got patient-derived cells that had reduced amounts of insulin, and they had a greater number of cells that were positive for both insulin and glucagon. So again, consistent with PAX4 playing an important role in repressing this alpha cell signature with the glucagon. So one of the beauties of being able to study a transcription factor is that RNA-seq and transcriptional outputs are a really rich data set for understanding how the transcription factor might be playing a role in regulating its expression of genes within the pancreas. So we took a pretty large study. We did 12 individual human iPSC lines in triplicate, and we looked at them at 4 different stages of beta cell development. So this is a total of 144 cell lines. And we differentiated them along the same path of 7 different points, taking these 4 points and then sequencing them. And what I want to point out is that the greatest difference we've seen across the cell lines is due to the time of differentiation. So that makes sense. The cells that are undifferentiated, the iPSCs all cluster together, and those that are endocrine progenitor cells cluster, the islet cells cluster, and the pancreatic endoderm cluster. And if we look at what the differences in gene expression at these across the different genotypes, we see that the greatest difference is at the endocrine progenitor stage. And again, this makes sense because it is where we have the highest levels of PAX4 expression. And if we take those genes that are differentially expressed at the endocrine progenitor stage and look at some of the pathways and processes that are involved, we see that they, a lot of them make a huge amount of sense. We see defects in metabolic processes. We see defects in cell protein metabolic processes. But intriguingly, we also see a defect in the cell response to stress. So this prompted Adrian and his team in Singapore to look into this in more detail and to perform seahorse experiments on the differentiated cells to see what their behavior was like in terms of their glycolytic function and mitochondrial function. So you can see here that Adrian was able to demonstrate that there was a bioenergetic switch from glycolysis. So you can see that here we have significant differences in many of the outputs of the seahorse experiment for glycolytic activity. And then when we look at mitochondrial respiration, you can also see that there is a switch to oxidative phosphorylation in the cells that harbor the type 2 diabetes variant. So perhaps this is explaining the finding from our transcriptomic analysis that there is an effect on stress, because we're certainly seeing the behavior of our cells is different when studied by the seahorse experiment. So one of the things that you can then do with CRISPR, which is a really powerful way to show whether or not something that you have discovered is attributable to the DNA change, is that you can use CRISPR to correct the spelling mistake in the DNA, to correct the mutation, and you can look to see whether or not this rescues the phenotype that you've observed. So we were able to take the patient-derived cell lines and to use CRISPR to correct the variant, and then to redo our experiment. And when we did this, we were able to show that we could rescue by the number of bihormonal cells, and we could increase the amount of insulin content. So this shows that the insulin reduction and the increased number of double positive cells is attributable to this change in the DNA sequence in the PAX4 gene. So why does this single nucleotide change that swaps the arginine to a histidine, why does this alter the function of the PAX4 protein? Well, we thought we would switch now from a stem cell model into endo-CBTRH1s and firstly see if in a mature model of beta cell function we could see effects on insulin secretion. And we did this by 2 different approaches. We did it by a transient reduction in PAX4 expression and by making a stable cell line with using lentivirus to reduce PAX4 expression. And with both of these independent experiments, we were able to show that when you lose PAX4, you have reduced insulin secretion and reduced insulin function in a fetal-derived human beta cell line. We then used the same cell line, endo-CBTRH1, to study the effects on gene expression using a luciferase reporter to see whether or not the mutant variety of PAX4 was able to regulate the insulin and glucagon promoters correctly. And we were able to show in this human beta cell model that the type 2 diabetes risk allele had evidence of increased glucagon promoter activation. So again, explaining and supporting why we would see increased levels of glucagon transcript and protein in our cells from Noel.
Yes, we have another question from Klaus. Why is the glucose effect on insulin secretion in the control cells so minimal? In human primary islets, it is at least tenfold.
Why? Say that one more time. Sorry.
Why is the glucose effect on insulin secretion in the control cells so minimal?
Oh, from the endos. That is what's reported from this cell line, Klaus. It's typical to only have a small effect in this fetal cell line to glucose. That's the way it works. And even the more recently reported lines, version 3 and version 5, still have a fairly minimal insulin secretion response. Okay. Okay. So I've shown you now how we've been able to show with luciferase assays effects of the mutant PAX4 protein on the ability to repress the glucagon receptor promoter rather in beta cells, which tallies very nicely with the fact that we see increased expression. So let me summarize this first part of the talk. We've studied the impact of this R192H variant in the PAX4 gene that's found exclusively in the South Asian population. We've studied it in individuals who carry the variant. We've studied it in isogenic and patient-derived iPS cells and in human fetal-derived endo-C-beta-H1 cells. We've shown that the type 2 diabetes risk alleles reduce beta cell function in vivo and lead to a de-repression of alpha cell genes in vitro. And collectively, we think this shows that PAX4 loss of function alleles increase diabetes risk by altering endocrine cell development. So I think this supports a very similar, if not identical, role for PAX4 in EVA. I actually saw the hand raise this time. Did you have a question?
I think I have to unmute her. Hold on one second. We've never done this as a webinar. Sorry for the changes. There you go. Eva, go ahead.
Okay, so I guess I'm going to have her slow down just a second. Okay, so the loss of function, we think, alters the balance between glucagon and insulin. Is that what you're trying to say?
Yeah, so
oh, good, perfect.
Yes, so we have more cells that are making both insulin and glucagon. So your beta cells aren't doing exclusively insulin.
Thank you.
You're welcome.
Good.
Okay. Was there another question?
I just said fabulous. Thank you.
Okay, thank you. Right, so now I'm going to shift into the second part of the talk, which is going to deal with moving from working on just one locus to going genome-wide. And I'm going to tell you about our efforts to look at the genes that make insulin and how we've integrated that data with type 2 diabetes genetics. So this is data that was generated by a really fantastic PhD student in the lab at the time, Antje Rotner. And what Antje did was use a genome-wide CRISPR library, the Toronto knockout library, to ask the question, what happens if I remove each gene in the genome from a beta cell to the amount of insulin the beta cell is able to make? So you set up the experiment so that you put guide RNAs that will knock down just one gene into one cell, and then you grow up the cells and you use cell sorting to separate them out into populations that have either a small amount of insulin or a lot of insulin. And then we can extract DNA from the populations of cells, and we can map back the RNAs for each of the genes to work out whether or not this population of cells that has little insulin, which genes are responsible for that, and which genes are responsible for having lots of insulin. So this is a difficult experiment to do because the cell line, this human beta cell line, is very, very slow growing. It only divides about once a week. So we only split the cells once a week. And it takes us about 4 months to collect the required number of cells to be able to do an experiment of this size. We needed 700 million cells to be able to do this in duplicate. So this is Antia spending enormous amounts of time at the tissue culture hood. At the 4-month period when we had the 700 million cells, it would be a very long day working on them at the hood. But fortunately, this experiment worked, and it worked really well. And across the 2 replicates, we were able to identify a total of 580 genes, which we feel we have robust evidence that they influence the amount of insulin in a pancreatic beta cell. So this has been a really fantastic collaboration with both Carl Galton and with Maria to be able to make this data that we've generated available to as many users as possible. I've put a QR code up here because Maria has made the data available on the Type 2 Diabetes Knowledge Portal and the Common Metabolic Disease Knowledge Portal. You can basically go in and you can type in your gene and you can look to see whether or not it is one of our 560 hits. You can also download the original data, reanalyze it. It's all there for people to dig into and explore. I'm just going to take a moment to say that we were also able to work with Maria to go back to a paper that we published in 2016, where we took 300 genes at 75 GWAS loci for 5 different cellular phenotypes and studied their impact on insulin secretion and content. And Maria and her team have also made these data available on the portal. And here's a separate QR code that will take you to that rich data set to explore. And as you can get a sort of sense of down the bottom here, these are how much insulin is at low glucose, high glucose, stimulated with IBMX or with tolbutamide, and also effects on cell count. So a really rich data set for people to explore. Okay, so back to the science. So we intersected the 580 genes that Antia had identified with a total of 336 predicted type 2 diabetes genes that we took from a curated list from the knowledge portal. And the intersection of these 2 lists gave us 20 effector transcripts where we had evidence from our cellular phenotyping that their loss affected the amount of insulin in a beta cell. And we chose to focus on one where there was intermediate evidence for a role in diabetes risk. The strongest evidence came from an independent signal, a coding variant in CalCoco2 that had been picked up in a proteomics study, but there was absolutely nothing known about its role within the pancreatic beta cell. Most of the literature reported an effect in infection and an effect with mitophagy. So we were really curious as to know what CalCoco2 could be doing in a pancreatic beta cell. So Ying Yi arrived in the lab around this time. She just completed her PhD with Erik Renström in Lund, and she was ideally placed to answer this question. So here are some beautiful EM images of the endo C-beta-H1 cells, either with or without CalCoco2. And the arrows are pointing to insulin granules. And what Ying was able to show was that when you lose CalCoco2, you get a reduction in the number of insulin granules, which totally fits with us seeing an effect on insulin content. But what was really fascinating was that this effect on granules seemed to affect more the immature granules rather than the mature granules. And if you look, you'll see that some of the granules look like dense black dots and others look more like fried eggs. And we saw that there was more of an effect on the fried eggs. So we were able then to look at the EMs and spot that there was evidence at a visual level of a process called autophagy, which is self-eating. So it looked as if we had parts of the cell machinery that were eating insulin granules. And Ying was able to demonstrate evidence for autophagy using IF. And you can see that here on the right-hand side of my slide. So we were able to demonstrate in this cell model that loss of CalCoco2 altered insulin granule homeostasis through autophagy. So since that publication, we've been really trying to get a better handle on what CalCoco2 might be doing in the beta cell. And we were curious about any function it might have within the mitochondria. As I said, it previously had been reported to play a role in mitophagy. And when we looked at our EM images, we could see that there was evidence for effects on the mitochondria because we saw these unusual mitochondrial cristae that were the wrong shape. So, and originally we had just measured ATP production in the endos and we saw no differences. But thanks to the arrival of Yonghyun Lee and her familiarity with the seahorse assay, we extended our work in this direction. And Yonghyun has been able to show that, like Ying Ying's experiments, there was no effect on ATP production, but she's seen evidence that there is an effect on maximal respiration and, importantly, on the spare respiratory capacity of the mitochondria. And if you think about it, this makes a lot of sense because if you challenge the cells with a greater metabolic demand, they won't be so able to step up to the task, which could affect their ability, therefore, to secrete insulin. So this is an area we're continuing to investigate and do more work on, but it looks to us as if there is a role for CalCoco, not just within granule homeostasis, but also with mitochondrial function. So let me summarize part 2 of the talk. I walked you through a genome-wide loss-of-function CRISPR screen in human beta cells for insulin content. I showed you that we'd identified 560 genes which influence insulin content and that 20 of them are potential effector transcripts at GWAS loci. You can find the gene list here on this QRL. And I showed you that we'd been able to establish that CalCoco2 is a novel regulator of insulin homeostasis through autophagy. Okay, so I'm going to finish up with some unpublished data on our recent efforts to use a unique islet resource that we've been privileged to help establish with Patrick McDonald from Edmonton. And where we've now performed a GWAS in a dish to identify novel regulators of insulin secretion. So let me introduce you to the wonderful team that have helped to put this resource together. First up, the team from Alberta, led by Patrick McDonald. This is a wonderful group of scientists who have painstakingly over many years isolated the islets and performed the functional characterization of them. We've been collaborating most recently with Jim Johnson and Yelena Kovac from UBC. Jim has also been working on the same islets from Patrick and has put down a couple of really valuable data sets that I'll describe shortly. We have a long-standing collaboration with the former postdoc, Jason Torres, back in Oxford. The team here at Stanford, who are responsible for the genomic element of our investigations. And a long-standing collaboration with Andrew Morris in the UK, originally in Oxford, now in Manchester, who is an expert in genome-wide association studies and statistical genetics. So we have used this resource for many years to support findings that we have identified either in rodent models or in beta cell models. And we've been able to use the resource that Patrick has established to demonstrate that we see replication of effects in model systems in human primary material. But we've been waiting for that moment when the numbers became large enough that we could try and leverage this to identify novel variants and mechanisms for human islet cell dysfunction by going genome-wide. So the resource and multi-omic toolbox that we've been able to work with, that Patrick has really led, has been the isolation of islets from human donor pancreases, which his lab have performed detailed cellular phenotyping on with static incubation, patch clamping, and then most recently Jim Johnson's lab has also done perifusion on a large number of these same donors. Our lab, originally in Oxford and now at Stanford, has done the genotyping of all donors. And we've also contributed to putting down omic data on the resource with chromatin state, with bulk ATAC-seq and bulk RNA-seq. And then most recently, Jim has used mass spec to put down a proteome on a large number of these donors. So this is a really valuable and rich resource that we've been able to interrogate for this part of our project. So here's a study overview. We've just completed the first round with a GWAS on 329 donors. I won't go through the methods here, but we've had to pay enormous attention to how we do this given the limited sample size and the high possibility of false positives. So we have taken replication very seriously, looking for replication in both islet perfusion data and in exocytosis. We've been even more cautious to make sure that things line up by using a method developed by Jason to show that there was evidence for action in human islets. And then we've integrated our data with very large omic data sets. We've got just over 800 RNA-seq donors, 223 ATAC-seq donors, and mass spec proteomics on 138. Over here are the details of what's unpublished and what's published in case people are interested. We've also been able to work with wonderful data from Kyle and from Klaus and from Jorge Ferrier to look at the looping of promoters and enhancers within islets. And then we've been able to do fine mapping and co-localization using a variety of valuable genetic data sets produced by many of our collaborators. So this framework is really about being able to be confident that the findings that we reach are backed up by other data sets and not exclusively drawn from what is a small GWAS study. So here are the results. This is a beautiful circular Manhattan plot. On the inner ring, you're seeing the results for insulin content. On the outer side in dark pink, you see the results for insulin secretion, the chromosomes around the outside in black. And you can see some of our notable loci in black and in brown. So this has all been done by a very talented postdoc in the lab, Seth Sharp, who set up this pipeline and done all of the analysis. And as you can see, Seth has identified one signal that reaches a very stringent genome-wide level of significance, and 18 at a slightly reduced threshold for GCIS at high glucose. And a larger number of signals have been identified for total insulin content. We've been able to go into our replication data sets, and we can replicate 10 of these for insulin secretion and 15 for insulin content. So I want to draw your attention to a few things on this chart. First of all, 6.2 and ADSI5, which are two well-established GWAS signals for fasting glucose and type 2 diabetes. And then a novel signal, PANX2, which I'm going to spend the next slide telling you a little bit more about. And just a reminder that these are like all GWAS studies have been labeled with the nearest gene, so don't take it as the effector transcript to go and get excited about until there's more evidence to do so. So let's dig a little bit more into our top hit, which is the PANX2 Soleno locus. And here you can see a deeper dive into the genetic evidence, the lead variant here in purple and the other SNPs that are in high LD. If we look at the effect of having this genotype, which has a really attractive minor allele frequency of 26%, making it quite easy to detect in a small, modest sample size, you can see that for every allele you have a 5,460 picogram per mil difference in your insulin secretion. Jason's method supports a role for this locus in all tissues, but importantly in islet. And you can see that we have the very rare feature here of having co-localization of this signal with an expression QTL for how much gene you make in an islet, chromatin accessibility, and a protein QTL. This is really remarkable to see this level of concordance across data sets. And if we go to the portal, we can see that there's also evidence for association with HbA1c and for fasting proinsulin. So lots of evidence to support this being a genuine finding. So this is how the functional genomics lines up at this locus. Thanks to Han Sun for assembling our own data and data from many wonderful other resources, many of them made by members of our consortium. So you can see that the top, the 10 SNPs in the credible set, and you can see here highlighted in orange how one of our credible set SNPs lines up over open chromatin in bulk ataxic. It sits in an enhancer from Jorge Ferrer's data. From Kyle's data, we can see that it's in open chromatin in alpha cells, in beta cells, but not alpha cells. And from data from both Klaus and Struan Grant and from Jorge Ferrer, we can see evidence that this is sitting in regions of the genome that are looping to regulatory sequence. And taken together, we've put together a schematic to show how this all comes together. We've got evidence that the variant that is associated with altered insulin secretion in a dish is responsible for increased amounts of the transcript of seleno, decreased amounts of an antisense RNA at the same locus, and increased amounts of protein. And having more protein results in having higher insulin secretion at high glucose. So a wonderful example of soup to nuts of identifying a variant and really being able to be confident about the effector transcript and then showing its effect on a cellular phenotype that is appropriate for disease. So some of our next steps, we're expanding our data set through collaboration with other resources and increased data that we have collected. We're going deeper to do a much more comprehensive analysis of co-localization across our data sets. And Tamada, a new postdoc in the lab who was previously in Pat's lab in Edmonton and is now on her own independent fellowship with us at Stanford, is following up on what seleno does in a pancreatic beta cell. Is it located in the mitochondria? Does it affect mitochondrial function? And can we recapitulate the effects on secretion when we perturb gene expression? So to summarize this last part of my talk, I've given you an example of this wonderful large multi-omic human islet dataset that we've been privileged to be part of assembling. I've taken you through our GWAS in a dish and our robust pipeline for replication. And I've shown you how we've integrated genetics with multi-omic data to identify effector transcripts, which not only give us the protein that we want to study, but importantly, the direction of effect. So let's wrap up with take-home messages across the 3 different elements of my talk. So we started off, if you remember, looking at molecular signposts with coding variants that take you to the gene you want to study. And I told you about our work on PAX4 that's been recently published. I then moved on to talking about how you can accelerate our ability to understand effector transcripts at GWAS loci by working genome-wide with a genome-wide CRISPR screen. And then I wrapped up by sharing with you some early data from our ongoing studies to look at identifying regulators of insulin secretion by performing GWAS in a dish. So I'd like to wrap up by thanking the wonderful people that I had the privilege to work with who have generated a large amount of the data that I've shared with you today. The PAX4 team, particularly Huey and Nicole, Adrian and Yishong, Antia, Yukion and Ying Ying for the CRISPR screen, and then this wonderful team of investigators that I'm working on for the GWAS in a dish across these different institutions. My colleagues in the news team are part of the Accelerated Medicines Partnership, my wonderful colleagues at the portal. Here are the links to the portal data and also to the podcast. And thank you to all my funders, the donors and their families. Thank you very much.
Thank you, Anna. That was wonderful.
Thanks so much, Anna.
Yeah. I think we've been answering questions in the chat, and I think we have one comment from Klaus, which is a very good one. I think he commented about he said it was lovely studies for your GWAS in a dish, and he would love to see how it coincides with HPAP data, if we can validate with HPAP data.
Yeah, absolutely, Klaus. That is the plan. Those are running currently. We will keep you posted.
So I will unmute anybody if they want to raise their hand to ask questions. We only have a couple minutes left, but you know, I got to say that it's always awesome to have Anna and present, because I think, you know, when you think about the portal, we think about this resource that's to the worldwide community about, you know, common metabolic disease. What's great is when you have an actual scientist who is generating the data and the insights and the sort of the knowledge, to coin a phrase, that we represent in the portal, to actually hear directly from them and see how their work sort of filters through to the portal is really rewarding and part of my favorite parts of these webinars. So thank you, Anna, for presenting such a lovely set of stories. That was awesome. Are there any other questions from folks?
Can I ask a question, Anna? So the third part is awesome. That's really amazing. But then also I feel like a little underpowered probably at the moment, right? For like novel discovery. But then I wonder, like another way that this could be used, which I presume you guys are thinking about and have done, is like grouping known loci based on their patterns of association with these phenotypes. Like, is that fruitful? Because then maybe you need like genome-wide significant associations, but you could highlight like patterns across traits and stuff.
Yeah, absolutely. There's Seth gave a whole talk on this at the ASHG and was able to go into it in more time. And to be honest, I could have just talked about that. But we have looked at genetic risk scores and broken it down, and we see some really intriguing findings there that make a lot of sense. And just remember, the PANX2 locus actually doesn't come up as a type 2 diabetes locus. It's been picked up here, but then if you go back into the type 2 diabetes data, we're starting to see more evidence. So I do think they're very complementary. And the point about sample size, we have got data through other collaborations in Europe that are going to almost double our numbers. And then, as Klaus pointed out, we're doing the same analysis in IIDP and in HPAP. So I think we're going to be in a really wonderful position to link up. I like the story with the Seleno because
it's very cool.
I mean, you and I know how rare it is to be able to go all the way from a variant to a phenotype. And I remember when you did it with melatonin, how excited we were because there are so few examples.
I guess my other question is any of this data useful for interpreting type 1 diabetes risk loci? Because there's I think the mechanisms there are less well understood, and then also I think generally a question of whether physiology, to what extent beta cell physiology plays a role in T1D risk. So I presume that this is useful for answering questions like that as well.
Absolutely. So you'll see that, well, you may not have seen because I went through it quite fast, but you'll see that the co-localization is not only being done with type 2 diabetes and glycemic traits, it's also being done with the latest data from Cassie and Steve Rich for type 1. So we will look into things for type 1 as well as type 2.
Okay. We have larger GWASs if you want them for type 1 diabetes that are almost 1 million people now. So if you want those, then.
Oh, silly question. I would love that. We need to connect you and Seth. That would be amazing.
Sure.
Beautiful. I realize we're 1 minute over time. I thank everyone for staying. Anna, thank you. This was wonderful. Folks who recorded this, we'll make this available on our website and YouTube page. And if you want to find any of the data that Anna so lovely referenced, you can also go to the KP Labs tab on our portal, and you'll find all of those results and more in the SNP to Gene feature on the website. We will reconvene January 11th in 2024 for our first webinar. That'll be a what's new and what's exciting, because there's a lot of new portals coming online in 2024 related to different tissues of the body and for across Common Fund data sources. So we're very excited to be participating in that. And I want to thank everyone for your attention. And again, thank Anna and Kyle for coming today and emceeing and giving a lovely presentation. All right. Take care, everyone.
Happy holidays. Bye-bye.
Happy holidays. Bye-bye.